Today's video is going to be about Dbt Bet 2008 question paper part B section 6. Question number 76 to 78. A transcription factor Y is examined for its DNA binding ability and its expression in different cell lines. A specific DNA binding sequence has been determined and is used as a probe in electromobility shift EMS assay, and specific antibody against the TFI is used in Western blot analysis to check whether the subcellular localization of the protein and the EMS and Western blot results of TFI activity in the muscle cells and nerve cells are shown below. Here you can see electromobility shift assay. Here is Western blot assay. Okay, this is probe which has DNA binding capacity. Okay, and here is nerve cell, here is muscle cell. So the DNA binding sequence in different cell lines, that is muscle and nerve cells, were determined by using the DNA binding ability. Okay, so here in EMS assay, as you can see, the muscle cells is at the top, whereas in the nerve cells it is at the bottom. So muscle cells DNA binding site is joined with protein sequence. Okay, that is antibody against TFY is used. So this antibody plus TFY, that is transcription factor Y, is joined together in the muscle cells, and so that it becomes heavier, so it moves slower than the other thing. Okay, this is EMS, and here you can see in Western blot. In Western blot, the subcellular localization of protein is determined. So where the transcription factor Y is present in muscle and nerve cell, it is present in cytoplasm or nucleus is determined using Western blot analysis. So as you can see here, the both the muscle and nerve cells has TFY but at different location. Like you can see, it is present in nucleus in case of muscle cells, and in cytoplasm the TFY is present in cytoplasm in case of nerve cells. Now we can see about the questions. The EMS results suggest that the EMS is, this is electromobility shift. So the answer is the TFY binds to the DNA in the muscle cell, right? TFY bound so that it is heavier, okay, but not in nerve cell. Okay, it is same as probe. So at nerve cell there is no boundment. So TFI binds to the DNA in the muscle cell because when antigen TFI bounds to the DNA probe, it becomes heavier than the probe, right? So if protein bonds to the probe, it becomes heavier than the probe. So the unbound things will be at the bottom, as in case of nerve cell, but bound will be at the top as it is heavier than the other thing. Okay, so this is the answer. TFI binds DNA in the muscle cells but not in the nerve cell. So next one, question number 77. According to the results of the Western blot, so what does this interpret? TY, TFY is expressed both in the nerve and muscle cells, right? So they are expressed in muscle and nerve cell, but in muscle cell it is expressed in the nucleus itself, but in nerve cell it is expressed in the cytoplasm. Okay, so when specific antibody against the TFY is used, TFY is bound to the antibody, right? And it forms the complex which is expressed in nucleus of muscle cell and cytoplasm of nerve cell. So this is the interpretation of the Western blot. So next one, question number 78. It is observed that the TFI activity is significantly blocked in the nerve cells in comparison to the muscle cells. Based on these data, what could be most likely mechanism of regulation of TFI in the nerve cell? So, what happens? TFI activity blocked in the nerve cell. when compared to the muscle cell. Why? It is likely mechanism that transcription factor Y translocation into nucleus is blocked, right? So in nucleus there is no TFY. If it is there, there will be a band over here, right? So inhibition of translocation of TFY in the nucleus occurred. As observed above, TFI gets blocked in the nerve cell as TFI expressed in the cytoplasm of the nerve cell, right? So TFI activity gets blocked by inhibition of TFY translocation in the nucleus of the nerve cells. So, you have any doubt, comment me in the comment section below. I will be happy to answer you. Subscription if you like. Okay, thank you.